Yes, so we have finished a pre-fertilization event. So next is fertilization event. As the name indicates, fertilization. So there will be fusion of two gametes to form the zygote. Okay, so before fusing, so what happens here? There are a few many steps they perform, especially in angiosperms, especially in flowers they perform. So we'll study one by one. So before fertilization, there are several steps. So the first step is pollen pistil interaction. Okay, so far we have finished pre-fertilization event. So till the gamete transfer, it ends with the pre-fertilization event. So the next process is a fertilization event. As the name indicates, fertilization is the fusion of male and female gamete to produce the zygote. Okay, before fertilization, before fusion of the male and female gamete, so there are some different steps which they perform, especially by the angiosperms, that is flowering plants. So the concept or the changes are taking place now with respect to the gynoecium or pistil. Okay, so the first step is pollen pistil interaction. Okay, so it is nothing but the interaction between the pollen grain and the surface of the stigma. So we know already the structure of stigma. I'll draw it in a bigger way for the better understanding. Okay, so this is our stigma and style. Okay, so what happens during the pollen pistil interaction? So the pollen grains which is dusted on the surface of stigma should be recognized by the help of what? By the help of the surface of the stigma. Okay, because we have already studied, so there is some compatible incompatible interaction between pollen grain and the pistil, that is stigma part of the pistil. So why they undergo this interaction? Because sometimes to avoid the other pollen grains or different species pollen grains will come and settle, land on the surface of stigma. In such a type, we call that as incompatible pollen grain or wrong type of pollen grain. Okay, this stigma, what they do, they doesn't allow the pollen grains, such pollen grains, to germinate on the surface, so they avoid the fertilization. Or sometimes it is also called as self-incompatibility. The self-incompatibility which is performed by the stigma itself. Okay, so what happens? If the plant doesn't want or the flower doesn't want to perform self-pollination, okay, because of some inbreeding depression, they encourage cross-pollination. Within the cross-pollination, we have studied the outbreeding device called as self-incompatibility. In such condition also, there will be interaction, okay, between the pollen grain and the stigma of the pistil. Sometimes they allow the pollen grain to germinate, sometimes they doesn't allow. Based on some chemical interaction, they recognize their species pollen grains. The stigma recognize it's the same species pollen grains because of this interaction. So hence, within the fertilization event, the first step is very important. Our stigma recognize the correct type of the pollen grains where they allow not. Okay, so now just imagine this pollen grain is the proper type, okay, of that pistil species only. So during that, they interact and they recognize each other. After recognition, after interaction, so what happens? The stigma produces chemicals, okay, or some secretions on the top of that landing surface. Okay, sometimes this secretion is also called as nectar. Maximum they are made up of water and sugar. Okay, so next step is secretion from stigma. Okay, after the secretion, some secretions will go inside the pollen grain and pollen grain undergo germination process, that is pollen germination. What do you mean by pollen germination? So we know there are some gaps between the exine wall, okay, that we call it as germ pore. Okay, so we'll revise one more time the structure of pollen grain. Okay, this is the germ pore. This is our exine wall, outer wall. This is inner wall, and they contain one, the bigger cell, that is vegetative cell, VCL, right? Another spindle shaped small tiny cell called as GC, generative cell. You should elaborate it. Okay, so what happens? Maximum we have studied 60% of angiosperms, the anthers dehisces at the stage, the pollen grain disperse out at the stage of two-celled. Okay, so what happens when the angiosperms dehisces? Okay, in the angiosperms, the anthers, when they undergo dehiscence, pollen grains are dispersed at the age of two cell, means which they contain one vegetative cell and generative cell. Okay, when these chemicals, when they go inside the pollen grain, so what happens? I'll draw here. Okay. From the pollen grain, like this, there will be emergence of pollen tube. So this is what we call it as pollen tube. The formation of pollen tube from the vegetative cell, the bigger cell, we call it as pollen germination. Where at this stage what happens? There will be vegetative cells in nucleus, and this generative cell, what happens? They undergo first mitotic division and form two male gametes, as we have studied three-celled stage. If you remember the gametophyte development, male gametophyte development, I have said you 60% of angiosperms, they dehisce the pollen grains or disperse the pollen grains at the stage of two cells, or sometimes at the three cells. So here we have taken two-celled example. They land, they absorb the secretions which is secreted by the stigma, and they form the pollen grain. When they form the pollen tube, they contain the divided two male gamete, and this nucleus of a vegetative cell will also follow this two male gametes. Okay, so this is the two male gametes. So this is the step. So what we can write? Pollen tube formation carrying, okay, two male gametes. Okay, so this is what. So next, the thing is, sometimes this style will be hollow, or sometimes this style will be solid. So there is difference. When the style is hollow, the pollen tube will emerge into the style. They travel into the style and they reach the ovary. Okay, but when there is solid style, there will be difficulty to travel. I'll tell you how they travel within the style, hollow tube, and they reach the ovule for the fertilization. If the style is solid, like how it is called as solid, this wall of style is made up of epidermis. Okay, epidermis layer, and within this epidermis they are composed of cells like this. Okay, a lot of cells, and these cells are called as conductive tissue togetherly. So if the style is solid, it will look like this. Okay, so what happens, the pollen tube, which contains a tube nucleus and two male gametes, so what happens, they need to travel within this tight conductive tissue. Sometimes there will be chance of rupture of this pollen tube end. So during that time, what happens, okay, so during this time what happens, the pollen tube, just imagine this is a pollen tube which contains two male gamete, and this is the vegetative nucleus. Okay, so what happens when they rupture in this solid style, okay, when they travel within the solid style, they got the chances for the rupture, okay, for the rupturing thing. So then what happens, they form a plug. Okay, this is a plug or compartment, okay, which is formed by some pectin chemicals. Okay, so this plug we call it as callus plug, which prevent the backward movement of all these three. Okay, backward movement they prevent, and one more thing they prevent by rupturing, okay, these pollen tube when they travel into the solid style. Okay, so next step is they travel through the style like this with the help of some chemicals, like there may be some boric acid also, sometimes a pectin, sometimes hydrolytic enzymes. So all these chemicals plays an important role during the travel of pollen tube into the style, because we know how the style, okay, how much this style is lengthy. Okay, from the landing position they need to travel, the pollen tube need to travel, and later they reach the ovule and they fuse, which form which perform fertilization. Okay, so next we'll start. So now pollen tube formation carrying two male gametes, they are traveling into the style, it may be hollow or solid. So now the pollen tube containing the two male gametes and vegetative nucleus enter into the ovule. Okay. I'll draw it much larger. Okay, ovary, style and stigma. So this pollen grain landed, interaction completed, and they even germinated also. Okay, so like this the pollen tube will be formed from the germ pore portion, which is containing now the two male gametes and what, vegetative nucleus. Okay, containing our one vegetative nucleus and the two main gametes. Okay, so this is what performed. Okay, so here I'll draw the structure of ovule. We know the structure of ovule. Okay, this is the structure of ovule. What ovule contains? Two layers of integuments, a nucellus region and embryo sac. Okay, just remember, this is a chalajal end and this is our micropyle end. So in angiosperms, maximum percentage, okay, in plants, the pollen tube the entry will be through micropyle end only. Okay, the entry of pollen tube. Such entry of a pollen tube from the region, micropylar region, we call such entry or the fertilization process, we call it as porogamy. Okay, sometimes it is through the chalajal end also. If it is through the chalajal end, it is called as chalajogamy, or sometimes through the integuments. Like, see, this is now entry through the micropyle. Sometimes there will be entry through the chalaja. Okay. Next, sometimes it may be they cross the integuments and they enter like this. Okay, that is through the integument sometimes. That we call it as mesogamy. So the syngamy or fertilization is of three types here. One, if they enter, the pollen tube enter through the micropyle end, porogamy, through the chalajal end, chalajogamy, or through the integuments, mesogamy. Okay, so we are going to study a porogamy type. Okay, so now which cells will show the route or pathway to enter through the micropyle end only for the pollen tube. Okay, ಯಾವ ಸೆಲ್ ಒಂದು pollen tube ನ ಏನ್ ಮಾಡುತ್ತೆ, ಆ pollen tube ಗೆ ರೂಟ್ ಅನ್ನ ತೋರಿಸುತ್ತೆ, ದಾರಿಯನ್ನ ತೋರಿಸುತ್ತೆ micropyle end ಇಂದ ಎಂಟರ್ ಆಗೋದಕ್ಕೆ ಓವ್ಯೂಲ್ ಅನ್ನ. Okay, so the cells here are, one is our egg cell. Remember the structure of embryo sac. Another two are our synergid, which contains finger-like projection called as filiform apparatus. At the chalajal end there are three cells called as antipodal, and the central cells contains polar nuclei. Okay, so the synergids, the filiform apparatus which is present in the synergids, okay, they show the pathway for the entry of the pollen tube within the embryo sac. Okay, I'll draw it in a bigger way. Mesogamy. Here I have enlarged the structure, ovule structure. So what happens, we know what is the ovule consist of. Embryo sac, and embryo sac are 7-celled, 8 nucleated stage. So 3 cells at the chalajal end called as antipodal cells, 3 cells at the micropyle end called as egg apparatus, where they contain 1 egg cell and 2 synergids. Within the synergids there is filiform apparatus, that is finger-like projection, which show the route for the coming pollen tube to get entered into the embryo sac. Okay, next is our, this is central cell, where they contain a polar nuclei, and we know already the ploidy of all are haploid, including male gametes, all are haploid in nature, but our polar nuclei or secondary nucleus are 2n, that is diploid. Why I have told you? Because They consist of two nuclei, but they are not fused. Each nuclei are n in number. So hence if they are present togetherly, hence it is called as 2n. Okay, yes. So the coming pollen tube, okay, so there will be a vegetative cell nucleus and two male gametes which is produced from the generative cell. Okay, this tube nucleus, what happens when they enter through the micropyle end? First they get entered into the synergids like this. Okay, the vegetative nucleus which is present here, they help to degenerate the end of the tube, pollen tube, and these two male gametes traveled within the synergids and deposited in the one of the synergid cell. Okay, from pollen tube they get traveled into the synergid. Now what happens? One male gamete fuses with the egg cell. Okay, this male gamete is N and our egg cell is N. Okay, this we call it as a pure syngamy. One male gamete fuses with one egg cell. Okay, which forms zygote, which is 2n. Now how 2n? N natured male gamete and N natured egg cell. So formed zygote is a 2n. So next, another male gamete will go and fuse with the polar nuclei or secondary nucleus. So this is also N and our polar nuclei is 2n, right? So what happens? The fusion of one male gamete and polar nuclei or secondary nucleus will form primary endosperm nucleus, or it is also called as PEN, pen, which is now 3n in nature. Okay, how it is 3n understood now? One male gamete which is N and the polar nuclei which is 2n. 2n plus n, 3n. So this step, there are first fertilization, this is second fertilization, hence it is called as double fertilization. Okay, double fertilization we understood now. And it is also called as triple fusion. Which step is triple fusion here? This one, the fusion of one male gamete with the polar nuclei, we call it as triple fusion. Okay, so hence in angiosperms the fertilization is of double fertilization and triple fusion. So first and second, so there are two fertilization, but one of the fertilization is triple fusion here, which is the product is 2n, 3n here. Okay, so double fertilization, triple fusion. Okay, we'll see how after the fertilization how the embryo sac will look. So this is our embryo sac. Okay, where this PEN, how it is formed? Here there is a PEN. Okay, this is the PEN which is 3n, and these antipodal cells, this is the zygote which is 2n, and these. Okay, so what happens to this? Antipodal cells get degenerated and even synergids also get degenerated. Okay, so the fate of the antipodal cells and synergids after the fertilization, double fertilization, triple fusion is they undergo degeneration. They completely vanished and the remaining thing is only primary endosperm nucleus and our zygote. This primary endosperm nucleus will give rise to endosperm. Okay, remember it is always 3n because our PEN is 3n, and zygote will form into embryo, which is 2n, and the development of endosperm and development of embryo is our next event, that is post fertilization event. Okay, this is what the fertilization event. Okay, understood? So it is very important to find out which male gamete goes and fuses with which cells in the embryo cell. Remember, always they consist of two male gametes. How the two male gametes are formed? By the generative cell, which is present in the pollen grain. So what happens? One of the male gamete fuses with the egg cell to form zygote. Another male gamete fuses with the polar nuclei or the secondary nucleus to form PEN, primary endosperm nucleus. So, 2 times they undergo fertilization. Hence, it is called as double fertilization. Why triple fusion? This step, the second fertilization, what we call, that fusion is nothing but triple. They form 3 diploid staged structure, that is PEN, which is in turn later forms endosperm. Okay, endosperm is the thing which gives nutrition for the developing embryo. Sometimes endosperm will persist, sometimes may not. Like in some seeds, okay, when we see the seeds, at the end we only find the cotyledons, like in groundnuts, okay, or all the cereals and pulses, we can see the cotyledons only. Okay, but example, when we go to coconut, the liquid part, the liquid portion, okay, of the coconut is nothing but nuclear or liquid endosperm, we call it as. That is one of the endosperm which helps or which gives a nutrition to the developing embryo. Okay, this we are going to study in the last event, that is post fertilization event. So hence in angiosperms we find special kind of fertilization, that is double fertilization, triple fusion. Okay, what is the fate of antipodal cells and synergids? They get degenerated after the fertilization process. Understood? They may ask the ploidy of all these things. Remember, endosperm is always 3n. Our embryo or zygote is always 2n. Okay. Later the embryo will form seed. Okay. Understood? So this is what the fertilization event in angiosperms. Thank you.